Hi guys, welcome back. So today we're going to be discussing a few of the models associated with colorectal cancer. So colorectal cancer is a major killer especially in Western countries. There's generally two main pathways that we talk about when we're talking about how colorectal cancer develops. And we're going to look at these pathways in the context of two clinical syndromes: familial adenomatous polyposis, also known as FAP, and Lynch syndrome. Before we dive into that, I think it's really important that we revise this APC beta-catenin kind of pathway and relationship. So APC is a tumor suppressor gene, and beta-catenin is a protein that's usually driving cell growth. And what happens in a normal cell is APC is involved in the formation of this destruction complex, and the destruction complex binds beta-catenin and destroys it, preventing it from entering the nucleus and driving cell proliferation. Normally, when Wnt signals, Wnt's this little molecule right here, when it signals, it tells the destruction complex to dissociate, and as it dissociates, it allows beta-catenin to then enter the nucleus, bind the DNA, and then result in cell proliferation. What we see in a lot of these cancers is there's something wrong with our APC gene, and that means that we don't get formation of the destruction complex, so beta-catenin is free to constantly enter the nucleus and drive proliferation. And as we know, tumors are just uncontrolled cell growth. So this high levels of beta-catenin in the cell allows uncontrolled cell growth, tumorigenesis, and eventually cancer as well. So moving on then to our first kind of model. So familial adenomatous polyposis is fairly rare, but it's very significant. It results in 100% lifetime risk of developing colorectal cancer, and I'll tell you, and you can see why. So this here is just a normal colon. You can see the mucosa is nice and smooth and shiny, and then if we scroll down a little bit, this is the colon of someone who had FAP, and you can see all these little dots here. These are all polyps, and in the colon, polyps are basically precursors to cancer. And so you can imagine, often if people go get a colonoscopy or whatever, they might find one polyp or potentially two. They'll remove them, and usually if it's a pedunculated polyp, so kind of this is the wall of the colon, it kind of comes up like this. Usually they'll just put a little sling around that, tighten it, and then remove that polyp, and that person's cured, they'll be fine. If that polyp's allowed to continue to grow, it can invade down, and if it penetrates the muscularis mucosae, then it can become malignant, which obviously is really bad. And you can imagine in a case like this, you've got thousands of polyps all through the colon. And what that will result in, so you can see the colon starting up here with the appendix, just there. Each of these polyps has a chance of becoming a malignant cancer. And so this person is basically, you just can't remove all of these polyps, and they're constantly growing. So even if you remove them, they'll just keep growing back. So a person, if they go untreated, will have 100% risk of developing colorectal cancer at some point in their life, and they're very likely to develop it early on in life. Because of this, often these people will undergo a prophylactic surgery to remove their colon, and so they get a bag stuck on instead, which acts as basically a storage vessel for the feces that comes through from the small intestine. So this is quite a serious condition. It's a result of our first pathway, which is chromosomal instability. Chromosomal instability. And we'll compare that later on with Lynch syndrome. As you can see here, so what this is, it's a chromosomal instability within the APC gene itself. So there's actually something intrinsically wrong with the APC gene. It's an autosomal dominant condition, and because it's involving this APC gene, this gene isn't involved in only colon cancer and colorectal cancer. It's very much associated with a lot of tumors in a lot of different locations. So people with FAP are also at high risk of fibromatosis, upper gastrointestinal tract tumors, sorry, getting tongue-tied, and also brain tumors. And as you can see, there are thousands and thousands of polyps. So this follows the first model, which is that chromosomal instability. You've got something directly wrong with the APC gene. The next one that we see is Lynch syndrome. Now, Lynch syndrome, it's very hard to find a photo. It doesn't look like this. In fact, it's usually, most of their mucosa will look like this, but they might have one polyp, or if they're unlucky, perhaps 2 or 3. The biggest thing is that those polyps develop a lot earlier in life than what you'd expect with anyone else. Lynch syndrome is again autosomal dominant, and it's due to this microsatellite instability pathway. So generally it's due to a defect in, it's due to defects in mismatch repair genes, and these are usually genes such as MLH1 and MSH2. What these genes usually do is, as cells replicate, they naturally accumulate little bits of DNA damage, and these genes, these DNA mismatch repair genes, go in, they create proteins which go in and they fix the problem before it becomes an issue. So, you get 1 or 2 bases that get mixed up. These genes fix those bases back to the way that they should be, and those genes don't get a chance to accumulate. In Lynch syndrome, you don't get this happening. These genes are dysfunctional, and that means that instead of clearing up those little mutations as they happen, those mutations accumulate and accumulate and accumulate, and they just keep accumulating. Eventually, you'll get those mutations happening in genes such as APC, and because of that, you start to get that uncontrolled cell growth in those polyps. But because it's kind of an indirect pathway towards APC, you only get a few of those polyps happening. You don't get the thousands and thousands and thousands. As I said, so only a few polyps. With Lynch syndrome, because it's affecting these genes here, these mismatch repair genes, you actually have a much higher risk of cancer in a whole range of systems. So we see in colon cancer that you're much likely to have it at a young age, early onset. So 50 is generally, getting colorectal cancer before the age of 50 is definitely considered young. Usually we say before 60 is early onset. And by age 70, a very large portion are going to have colorectal cancer. And I don't know if you can see that there, but in the general population, 0.2% will have colorectal cancer by age 50. So more than 25% is a huge increase in risk. And by 70, only 2% in the normal population. So in Lynch syndrome, there's an 80% increase in risk, which is rather insane. And as we say, those risks are mirrored across a whole bunch of different organ systems. So in women in particular, it's a major cause of uterine cancer and ovarian cancer. And so as I say, rather than, as opposed to FAP, Lynch syndrome is affecting microsatellite regions, results in much less, only a few polyps, but they still have a very high risk. For these guys, removing the colon is still an option, but often what they'll do is they'll get much higher screening frequency and things like that for these polyps. Because there's only a few of them, they're able to be removed a lot easier. And while they will grow back, again, you're not having thousands growing in a sheet. There's just going to be a few that can be easily removed. So these guys get screened for cancer very regularly from a young age, in an effort to try and, identify any tumors that arise early. And and as you can see as well, as opposed to FAP, which only kind of had a few extra risks associated with it, Lynch syndrome is very much associated with extracolonic manifestations of the disease. Okay, that's today's lecture. That was quite a short one. If you've got any questions, please feel free to leave them in the comments below. And if you've got any ideas for more videos you'd like me to do, or if I haven't explained anything well, I know this is quite a superficial video. We didn't dive into too much of the genetic details. But if you'd like me to explain anything in more detail, please let me know in the comments below.